All right, let's jump right in. Have you ever wondered why we even bother studying microscopic cells? I mean, it is so easy to just picture biology as nothing but dusty textbooks and really abstract diagrams, right? But the reality? The microscopic world is actually the front line for curing diseases, engineering life-saving medicines, and literally feeding a rapidly changing planet. So today, we're going to map out exactly why studying cells is the master key to solving these massive global issues. And by the end of this explainer, you're going to have a crystal clear, step-by-step roadmap for our entire journey into the microscopic universe.
First things first, let's talk about the word cytology. Sounds a bit intimidating, right? Well, if we break that down into its Greek roots, we immediately strip away all that heavy scientific jargon. Ketos simply translates to a hollow container, and logos means the study of. So cytology is really just the study of these tiny hollow containers that hold the entire machinery of life. But listen, today this science goes way, way beyond just squinting through a lens. It is a highly active, incredibly dynamic field.
Because here's the thing, cell biology isn't just about describing little microscopic blobs. It is the absolute foundation for solving major, real-world problems. We can basically break this down into four main pillars: medicine, genetics, agriculture, and biotechnology. Think about it like a giant puzzle. You can't fix a complex machine if you don't fundamentally understand its parts. And in the machinery of life, the cell is that foundational part. So let's look at how this actually plays out in the real world, starting with disease.
Take cancer, for example. There's this really common misconception out there that it just broadly attacks the whole body all at once. But in reality, cancer begins in just one single cell that loses its internal control mechanisms. It literally forgets how to stop dividing. And this right here is exactly why a deep understanding of standard, normal cell division is absolutely crucial. We have to know what the healthy process looks like so we can pinpoint the exact moment things go rogue.
And understanding that rogue behavior is exactly how doctors are able to target the disease without harming the patient. Because researchers understand cellular division, they can engineer chemotherapy drugs to act like highly trained molecular hunters. These drugs are specifically designed to seek out and destroy cells that are dividing way too quickly, which, as we just talked about, is the hallmark of that rogue cancer cell. It's a direct, tangible application of cytology. Without knowing the mechanics of how cells divide, we simply wouldn't have the drugs designed to stop them.
Okay, let's switch gears a bit within medicine and compare a human cell to a bacterial cell. Take a look at this crucial structural difference. A typical human cell, the exact kind making up your body right now, has a soft, flexible outer boundary. But a bacterial cell, it features this tough, rigid outer layer known as a cell wall. It acts almost like a microscopic suit of armor. And if you've ever recovered from a nasty bacterial infection, this one single structural difference is exactly what saved you.
So, how do we use that to our advantage? Well, medicines like penicillin are uniquely calibrated to recognize and destroy that specific bacterial armor. And because our human cells just don't have a cell wall, the medicine completely ignores them. It targets the bacteria, breaks it wide open, and destroys the threat, all while leaving you perfectly unharmed. That precision comes entirely from understanding cellular architecture.
Let's pivot over to biotechnology, where this foundational knowledge is literally creating life-saving insulin for diabetics every single day. Back in the day, insulin had to be slowly and expensively extracted from pigs or cows, but today, it's an elegant three-step process. Because we understand how cells read DNA, scientists can literally insert a human insulin gene directly into a bacterial cell. The bacteria reads it as its own and transforms into a microscopic factory, mass-producing pure human insulin. We are basically programming cells to save human lives. How wild is that?
Now, in agriculture, scientists are doing something equally amazing. They use a technique called tissue culture to grow an entirely new plant from just a single piece of tissue. By placing one single cell from a leaf in a lab and feeding it the perfect blend of nutrients and hormones, it regenerates into a complete, fully formed plant. This means that if we find a plant with an absolutely superior trait, we can isolate its cells and replicate it flawlessly over and over again.
And the real-world result of this? We're talking game-changing innovations like disease-resistant bananas and high-yield rice. Faced with a rapidly growing global population and totally unpredictable shifting climates, agricultural scientists have actually engineered crops to produce massively higher yields and withstand devastating fungal diseases that could otherwise wipe out our entire food supply. The security of the food on our tables literally relies on studying cells.
So, now that we know the massive stakes here, let's lay out your roadmap for this chapter. Having a structured guide is going to make it so much easier to see how all these tiny biological pieces fit together to form the big picture.
Over our next 5 blocks, we'll cover all the essentials. Block 1 is all about techniques, like microscopy. Block 2 zooms in on the cell wall and membrane. Block 3 explores the cytoplasm and organelle functions. Block 4 breaks down the difference between prokaryotes and eukaryotes. And finally, Block 5 uses bacteria as a model for prokaryotic structures.
In our very next explainer, we'll be kicking things off with Block 1, exploring the practical, real-world tools that scientists actually use to see and study these microscopic containers.
But before we officially wrap this up, I want to challenge you real quick. Take a second, pause, and actively answer the question on your screen. Name one real-world application of cell biology and explain why it requires knowledge of cells. Think back to the cancer treatments, the penicillin, the insulin factories, or engineered agriculture. Testing your memory right now is, hands down, the best way to make this knowledge stick long-term. Just remember, every major breakthrough in the life sciences, everything that changes the world, starts infinitely small, right at the cellular level. Think about that, and I will see you in our next explainer.